Today we're going to talk about spermatogenesis. Spermatogenesis is the form of meiosis that males use to create sperm. Spermatogenesis happens in the testes. About 100 million sperm mature every single day in males. However, this doesn't happen from birth, it starts at puberty. Certain hormones like testosterone are starting to be released, and that's what triggers the beginning of spermatogenesis. However, males continue to do spermatogenesis for their entire lives. However, as a man gets older, spermatogenesis may be less and less efficient, producing fewer sperm. Now let's get into the actual steps of the process. You'll find that this is fairly simple compared to what we're going to talk about later, which is oogenesis, the process of making eggs in females. Spermatogenesis starts with spermatogonia. Spermatogonia are stem cells that are found in the testes. These stem cells can be used to make sperm. However, if the male continues to use all of his spermatogonia to make sperm, he will eventually run out. In order to prevent that from happening, his spermatogonia have to undergo mitosis to replenish that supply of stem cells. What happens is these stem cells divide. One of those cells that are produced, one of the daughter cells, goes back to become the new spermatogonia, and the other daughter cell can actually continue on through the process of meiosis to become sperm. The one cell that goes through spermatogenesis is going to be called the primary spermatocyte. The primary spermatocyte is also called the immature sperm. That's what we're going to call that from here on out. This primary spermatocyte is 2N or diploid. Because it's diploid, that means it has pairs of chromosomes. So in humans, we would consider there's 23 pairs of chromosomes, one maternally derived and one paternally derived. This immature sperm is then going to go through meiosis 1. Now we know that during meiosis 1, crossing over occurs. So that means that a small section of this chromatid is going to exchange with this chromatid. The non-sister chromatids within a homologous pair are going to exchange information. What also happens during meiosis 1 is that the two homologs separate. So one of each pair will go into one cell, and the other of each pair will go into the other cell. So therefore, our resulting secondary spermatocytes are going to be haploid, meaning they only have one copy of each chromosome. These secondary spermatocytes are then going to go through meiosis 2. However, you can see, actually, before we go on to there, you can see that these chromosomes are still replicated. The chromatids have not yet been pulled apart. Only the homologous chromosomes have been separated. You can see now that the secondary spermatocytes are going to go through meiosis 2. During meiosis 2, this is when the sister chromatids get pulled apart, and each new cell, which is now going to be called a spermatid, contains still haploid information, so they still only have one copy of each chromosome. However, now they are unreplicated. You can also see that because of crossing over, each spermatid is different from each other spermatid. So we have, in this case, 4 different options. When we have 23 pairs all undergoing independent crossing over and independent assortment, you get many, many more options. This whole process from start to finish, the spermatogonia to spermatid, is going to take place within the testes, in a specific location called the seminiferous tubules. I'll show you a picture of that in a moment. Once the spermatids are formed, they start to become more specialized in shape. This starts to occur within the seminiferous tubules, and then the sperm are transported into a location called the epididymis, which is located right on top of the testes. Each testis contains its own epididymis, and that is where the spermatids become full-fledged sperm. They mature, they learn how to swim, they start making some of the enzymes and other things they'll need for their long journey to find the egg. This is a picture of a seminiferous tubule. It's a cross-section. So what you see here is along the outer edge is going to be where the spermatogonia are. As they start to go through spermatogenesis, they will divide, and as they get closer and closer to becoming full-fledged sperm, they move inward. So along the outer edge, we'll have the spermatogonia. Further down are the primary spermatocytes, the secondary spermatocytes, spermatids, and then all of these in the middle here are tails. This is an actual picture that I took of a prepared slide that we have of the seminiferous tubule cross-section. So on the outer edge, again, still we have the spermatogonia. As they're going through meiosis, they are slowly actually moving towards the center of this tube. It's a tube, and so in the middle is sort of this open area where the sperm are going to be transported up into the epididymis. And that's the end of spermatogenesis. We'll see that oogenesis is much more complicated in terms of timing and in terms of products that are produced.